So we're going to finish talking about the regulation of enzyme activity. So we discussed the broad overview of how enzymes are regulated, either by gene at the gene expression level or at the activity level. And we said that everything that's in blue here, okay, all the arrows that are in blue, deal with bioavailability. Is the enzyme present or not? Okay, so is it made? Is it degraded? Is it in the right area? And then everything that falls under catalytic efficiency is represented by these processes here. Okay, so these are things that are going to affect the kinetics. Let's see. Kinetics affected by binding or chemical modification. Okay. So now we're going to be a little more specific about those catalytic efficiency means of regulation. Okay. All right, so I introduced ATCase to you in class. And again, the quaternary structure of this enzyme is that it has catalytic subunits and it's got regulatory subunits. Okay, so it has 6 of each, but it has 2 trimers. Okay, so it's got C3 times 2. And then it's got 3 regulatory dimers, which are shown in the purple here. So how does allosteric regulation in ATCase work? Okay, remember that ATCase is responsible for pyrimidine biosynthesis. And so those are our C's and our T's and our U's. Okay? And so ATCase catalyzes what is called the committed step. So this is the point at which no intermediates are funneled off into other pathways. Everything from this step on leads to the synthesis of pyrimidines. Okay, so ATCase is regulated both in an activation sense and in an inhibition sense. So one of the pyrimidines that it is responsible for synthesizing, cytidine triphosphate, okay, will bind to the regulatory subunits and inhibit activity. So that's a feedback inhibition mechanism where the end product of your pathway is going to be inhibiting your committed step. Okay. And then the other way that it's regulated is by ATP. All right, so ATCase has T and R states. So the T state, when we talk about enzymes, it's a little different than when we talk about hemoglobin. Okay, the T state is the inactive state, sorry. It's the inactive state. So this is analogous to our low affinity for oxygen in hemoglobin. And then the R state is the active state. And so that's analogous to the R state in hemoglobin, where you have high levels of affinity for oxygen. All right, so when CTP is bound to the regulatory subunit. That induces the less active conformation in the catalytic subunit. Okay? But when ATP is bound, okay, that induces a change to the active R state. Okay? And then you get carbamyl phosphate and aspartate being combined to form N-carbamyl aspartate, which is the reaction that this enzyme catalyzes. Okay? So the ATP-CTP balance in the cell is really going to fine-tune how much ATCase activity you get in the cell. All right. So when we look at ATCase activity, okay, if we just do have the enzyme in the absence of any inhibitor or activator, okay, it has semi-sigmoidal kinetics because it is, as an allosterically regulated enzyme, it is also, there's cooperativity, so that when you have if we go back here, when you have the substrates binding to one subunit, all right, they induce the conformational shift to the R state, okay? So you do get this kind of cooperative sigmoidal curve, okay? When you have CTP, okay, that inhibits ATCase activity, and that shifts the curve to the right, again, because it gives you sort of a lower affinity for your substrates, as well as reduced ability to catalyze the reaction. Now, when you add the activator ATP, okay, that changes the shape of the curve, so it becomes more of an enzyme that looks like the behavior of a Michaelis-Menten enzyme. Okay, but it also, again, it shifts the affinity for aspartate. So that it is higher affinity, it shifts the curve left, and the reaction goes faster and produces product faster. All right, so now we're going to talk about covalent modification. Okay, so this can be, either adding or removing a functional group. So that's one way of covalently modifying an enzyme, but another way is proteolysis or hydrolysis of a peptide bond, okay, or more than one peptide bond, okay, and this is what we would call zymogen activation. Okay. All right, so let's look at an example of enzyme activation via proteolysis. Okay, so the term zymogen we use to refer to the inactive form of the enzyme, okay, that becomes active once a peptide bond is cleaved, right? So if we look at pepsinogen, okay, it's another enzyme whose job it is to, the mature enzyme, the active enzyme pepsin, is an enzyme that cleaves peptide bonds in other molecules. But to become activated itself, okay, you have to cleave this N-terminal portion of the precursor molecule. Okay, so pepsinogen is the zymogen form of pepsin, which is the active form of the enzyme. And so pepsinogen has this area on the N-terminal, okay, where you've got a portion of the peptide that actually lays down in the active site, and it blocks its own active site. So it's not active. So in order for this enzyme to be able to accept substrate, we have to clear this out of the active site. And so, the way that this gets activated is that the peptide bond that connects the N-terminal portion to the rest of the protein gets cleaved. This goes out of the active site. So now you have an open active site where your substrate can bind. And these aspartic acid residues are important in the mechanism of pepsin, and they are now available to catalyze the cleavage of the peptide bond. So peptin is an example of an aspartic protease. The HIV protease is also an aspartic protease. So we've talked about HIV protease before, because the HIV virus produces these long proteins that need to be cleaved at certain spots to produce the active forms of its proteins, right? And the mechanism of both of these uses two aspartic acid residues to do the chemistry, unlike a serine protease where you have that catalytic triad of the serine, the histidine, and the aspartic acid. Okay. All right, so pepsinogen is the zymogen form, and upon cleavage it becomes pepsin and it is catalytically active. Okay, so another enzyme that is produced in zymogen form and then processed by proteolysis to produce its final form is chymotrypsin, and this figure got cut off a little bit. Apologize for that, but the important parts are visible here. All right, so chymotrypsinogen. Okay, so chymotrypsinogen, okay, is the zymogen form that's made, and this is what comes off of is the translation product, okay, of the gene for chymotrypsin. Okay. Now, chymotrypsin, like pepsin, is a proteolytic enzyme. So again, you don't want to be chopping. You don't in the cell. You don't want to be chopping up all your enzymes all the time. Okay. So that's why a lot of proteolytic enzymes are activated by some type of covalent modification or zymogen activation, so that they're only used when one needs them. All right, so chymotrypsinogen, okay, gets activated by trypsin. Okay, so one of the things that I want you to notice is that there are a bunch of disulfides in chymotrypsinogen, and one of them is that you don't see connects two parts of the protein that are very far apart in space. So we're going to be cleaving peptide bonds, but some of the parts are going to be held together by these disulfides. Okay, so trypsin, all right, cleaves, trypsin. Trypsin, there we go. So chymotrypsin gets cleaved by trypsin at this spot right here, right before residue number 16. Okay, and then what that leaves you with, okay, is kind of two parts. Okay, one of which is called pi chymotrypsin. Okay, and then Pi chymotrypsin engages in autocleavage. Okay, so pi chymotrypsin has catalytic activity. Okay, and it, oops, hold on, I just dropped my pen. Find it. All right, there we go, sorry. All right, so pi chymotrypsin cleaves itself. All right, and it does it in two spots. Okay, so this little spot here, okay, is going to leave. It also cleaves it here, okay, on the other end, and this little piece is going to leave as well. Okay, but all of the remaining parts, okay, are going to remain connected, okay, by those disulfide bonds. Okay, so this is going to be your active chymotrypsin molecule. Okay. All right. So the other type of covalent modification that we can have is, again, adding or removing functional groups. Okay, so common things to add onto proteins, okay, are phosphoryl groups, methyl groups, acetyl groups, anylyl groups, or uridyl groups. Okay, the most common thing is phosphorylation of a serine, a threonine, or a tyrosine. Okay, so this is the most common modification for turning things on and off quickly. They're done by kinases, which use ATP as a phosphate source instead of inorganic phosphate. And then sometimes something is activated by phosphorylation, sometimes it's inactivated by phosphorylation. Regardless, in either case, if you want to change the activity back, if you turned it on and you want to shut it back off, you can use a phosphatase, okay, to remove the phosphate group. Okay, so here's just an example, okay, of an enzyme that is activated by phosphorylation, and so serine 14 is where it gets. That is the part that gets phosphorylated. You'll notice that active site, which I'm just going to color green here, is not very close to the site of phosphorylation. But remember what happens when you add a phosphate group. When you add a phosphate, so if you add a phosphate, you're adding a lot of negative charge. That is going to have a huge impact on the structure of your protein. So you're going to get a structural shift, and that's what's going to make the active site become catalytically active. Okay. All right. So here's an example of something that's regulated by phosphorylation. Okay, this is an enzyme called glycogen phosphorylase. Glycogen phosphorylase is basically, it's the enzyme that peels off glucose molecules in animals. So animals will store glucose as glycogen. So this is so animals store glucose as polymer. So lots of individual glucose units, and glycogen is not just a chain of glucoses, it's highly branched. Okay. So we store glucose that way, and when we want to use individual glucose molecules to go into glycolysis to start ATP production, we need to, cut one off of the chain, and glycogen phosphorylase is the enzyme that does it. It also phosphorylates the glucose so that it can go into glycolysis in the second step. Okay, so it's going to So the enzyme, okay, is going to phosphorylate glucose, but the enzyme itself is activated by dephosphorylation and inactivated by phosphorylation. Okay, so if we look at, these are enzymes that regulate our metabolic pathways. Okay, so insulin, okay, is a hormone that stimulates us to remove glucose, from the bloodstream, send it into glycolysis, and start using it. Whereas glucagon and epinephrine, okay, are enzymes that are going to kind of have a different effect on our metabolism. Okay, so they're going to stimulate something called phosphorylase kinase, okay, to activate glycogen phosphorylase. So the insulin, okay, is going to deactivate glycogen phosphorylase. So glycogen phosphorylase, it is inactive when phosphorylated, okay, and it's active. It's active when it's phosphorylated. Sorry. Let me erase this. This is going to be inactive without phosphate, okay, and it's active, when phosphorylated, okay, all right. So what's going to happen is, so insulin is the hormone that we have in our body when we've got glucose in the bloodstream and we want to take glucose out of the bloodstream, okay? The purpose of glycogen phosphorylase, okay, is to make glucose available in the bloodstream by taking it out of our glucose stores, okay? So insulin is going to be the hormone that we use to shut off glycogen phosphorylase, because it's an enzyme, again, that gets, it's a hormone that gets activated when we have glucose present and we don't need to liberate more glucose, okay? So insulin shuts off glycogen phosphorylase, okay, because sufficient glucose is present. But let's say you've been fasting and then you go try to do some exercise, okay, your body is going to need glucose to make ATP. So that's when glucagon and epinephrine are going to come in. So these ones, okay, are going to turn on glycogen phosphorylase by activating an enzyme called phosphorylase kinase. Okay, so activating phosphorylase kinase, which adds a phosphate to glycogen phosphorylase, activating it. Okay? So that the action of insulin causes the removal of a phosphate and conversion to the inactive form, and so glucose does not get peeled off of our glycogen stores, whereas glucagon and epinephrine activate an enzyme that adds the phosphate to the glycogen phosphorylase, and that then causes release of glucose molecules from glycogen into the bloodstream so that they can be used for energy. Okay. All right. So that's the end of our examples for enzyme regulation, and we'll go over the answers to some of the practice problems in a separate video.